Chapter 7, Genotype to Phenotype. Today we'll explore more and take a look at what's involved when it comes to protein synthesis. This is a detailed recreation of a cell. There is a lot going on in this picture. But don't get too overwhelmed. We're only going to look at the organelles involved in making a protein.
Here we go. Now we can better see the nucleus. The nucleus is where it all begins. It is the control center of the cell. If you will, DNA comes from the nucleus. At the center of the nucleus is the nucleosis. Here, ribosomes are made.
The ribosomes assemble amino acids into a polypeptide. A polypeptide is a string of amino acids ready to be formed into the shape needed. Once this polypeptide is assembled, it moves on.
The nucleus has a phospholipid bilayer, just like the plasma membrane of the cell. In protein synthesis, the DNA is transcribed to mRNA. Then it moves out of the nucleus and into the cytoplasm to the ribosome.
Once a ribosome leaves the nucleus, it may attach itself to the rough endoplasmic reticulum. As you can see, there are the tiny purplish dots that seem to be going through the blue maze. There is also a smooth endoplasmic reticulum, but the ribosomes won't be stopping here. We call it the smooth one for that reason. It doesn't have ribosomes attached to it.
Its functions are the synthesis of lipids, steroid hormones, the detoxification of harmful metabolic byproducts, and the storage and metabolism of calcium ions within the cell. Ribosomes go in, and proteins come out. These newly formed proteins next make their way to the Golgi apparatus. You could think of the Golgi apparatus sort of like a dry cleaners, in that the folding is done. Proteins get folded into usable shapes. They can also have other materials added onto them, such as lipids or carbohydrates. It helps process and package proteins and lipid molecules, especially proteins destined to be exported from the cell.